Hi, I'm Bob Lessig. Welcome to a short lecture on how DNA evidence is evaluated. We'll start by going over some basic terminology. Several acronyms are used here. The RFLP process can be used to distinguish individuals based on the sizes of very specific fragments of DNA. So, why do people have different length fragments? Sometimes mutation is involved. In many cases, there are short repeated regions of DNA, and individuals can vary in how many of those repeats are present in an individual. We'll look at how forensic data is evaluated. Usually, they look for an exact match in those situations. Paternity testing is a little more complex. Rather than exact matches, you would examine inheritance patterns. It's a bit like a puzzle. Here are some acronyms to be discussed. PCR is the polymerase chain reaction, a method to amplify specific regions of DNA. RFLP is restriction fragment length polymorphism. Many people became aware of it after the O.J. Simpson trial. It allows for detection of genetic differences in a gel which separates fragments by size. VNTR is variable number tandem repeat. That genetic feature makes DNA fingerprinting work well. What are we attempting to accomplish? The human genome is very large. We have to look at very specific regions of DNA. Those regions are chosen based on variability in the population and the ability to detect that variation. In the United States, most testing is based on 13 regions of DNA. It might not sound like a lot, but it would be very difficult for one individual to match an unrelated person at all 13 locations. With paternity testing, we don't look for exact matches. At each of the 13 locations, everyone has two alleles, one from the mother and one from the father. So it's more of a puzzle to see if a father could have provided the child's genotype. Restriction enzymes were first seen in the 1950s, when it was discovered that bacteria could find a way to restrict viral infection. It was later discovered that these enzymes cut DNA at very specific sequences of DNA. Those sequences are often, but not always, 6 nucleotides long. Daniel Nathans discovered that cutting genomic DNA with these enzymes yielded very specific fragments of very specific lengths. An enzyme that cuts a sequence of 6 nucleotides will, on average, cut DNA into fragments of about 4,000 base pairs. However, that's on average, because that sequence occurs irregularly. Some are larger, some are smaller. Most individuals have fragments that have the same size patterns, with a few exceptions. It's the exceptions that DNA testing exploits. Restriction enzymes revolutionized biotechnology, making genetic engineering possible. Nathans shared the Nobel Prize in 1978 and later became president of Johns Hopkins University. RFLP stands for Restriction Fragment Length Polymorphism. Polymorphism is a computer science term, but in the biological sense, it refers to differences, usually phenotypic, within a population. The length polymorphism refers to restriction fragments that can vary in size among individuals. How can the length vary? Well, let's look at the top sequence. The red triangles represent restriction sites, those specific sequences where restriction enzymes cut. There are 3 sites in this region of DNA. Now let's look at the middle one, individual B. A mutation or SNP at the restriction site would remove the enzyme's ability to cut there, so there are only 2 sites to cut this individual's DNA. Finally, individual C has a short duplication of a specific region of DNA. A labeled probe can be applied to find this fragment. A probe usually comes from a stretch of DNA where there is little or no variation in the population. That way the label would work on everyone. Note the size difference between all three individuals in the labeled fragment. Individual A has the shortest fragment. Individual C has a slightly larger fragment, thanks to the sequence duplication. Individual B has the really long one because an entire restriction site was removed. DNA fragments can be separated by size in gel electrophoresis. Larger fragments migrate slower through the gel. It's those repeated sequences that are most often the target of DNA testing. Variable number tandem repeats basically refers to short sequences that appear back to back in genomic DNA. At these particular locations, individuals vary in the number of repeats present. One chromosome might have 5, and another might have 9, for instance. Providing even more variation is that we all have chromosome pairs. For instance, humans have 2 copies of every chromosome. Let's pick one. Let's say chromosome 2. We all have 2 copies of chromosome 2, one inherited from the mother and the other from the father. So an individual might have 6 copies of the repeat on the maternal chromosome and 8 on the paternal chromosome. Another individual might have 4 copies on the maternal chromosome and also have 4 on the paternal. That's a lot of variation. You could start to see how matching 13 locations would be difficult. The PCR process was invented in the 1980s. That also revolutionized biotechnology, allowing specific regions of DNA to become amplified. Even in a very large genome, PCR allows scientists to make many copies of a very specific region of DNA. The process makes so many copies that the copied region becomes the dominant DNA in the mixture. We'll see how that helps the DNA fingerprinting process next. Here is a VNTR region. Note that the top sequence has more copies of the repeat than the bottom sequence. PCR amplifies the region between the primers. Primers are carefully chosen sequences with little to no variation within a population. Because the top individual has more copies of the repeat, its PCR fragment is longer and moves through the gel slower. The gel at the right shows the longer fragment at the top. One thing is misleading. It is correct to say that one fragment is paternally inherited and the other is maternally inherited. However, this process does not allow us to say which one is which. Note also that some individuals would have the same number of repeats on the paternal and maternal chromosomes. Those individuals would have a single band on the gel rather than 2. Okay, here's how it works. The forensic sample is on the right. Individual A, B, and C are suspects. The gel is on the bottom. Which one is a match? Note that this is just at 3 locations, not 13. 13 locations would give you a pretty good fingerprint. What regions are examined? In the United States, the FBI uses CODIS, or the Combined DNA Index System, which is pretty standard throughout North America. Other countries have different standards. The United Kingdom uses 11 locations. The CODIS system examines 13 locations. There are known frequencies throughout the US population broken down by race. The locations are spread out over the autosomal chromosomes, meaning not the sex chromosomes, X and Y. On the diagram, you see AMEL, A-M-E-L, on the X and Y chromosomes. That is used by the FBI to determine gender, but it is not one of the 13. Here is some data from one of the CODIS locations. The alleles in this case represent size differences. You can see that some alleles, particularly 11 and 12, are more common throughout each population. This particular location is an intron in the CSF1R gene on chromosome 5. See if you can find it in Ensembl or GenBank. So let's do a partial paternity test. We'll do it at 4 CODIS locations. The mother's genotype is in the left column, and the child is in the second column. 4 suspected fathers are listed in the final 4 columns. We'll start with row 1, the CSF1PO location. The mother has a 10 and a 12, and the child has 11 and 12. The 11 in the child could not have come from the mother, so the child must have gotten the 12 allele from mom. The 11 must have come from the father. Which males could have provided the 11? All but male number 1. He has 2 copies of the 10 allele. Male 1 cannot be the father. We will put in an X. The FGA region. The mother only has the 23 allele, 2 copies of it. The child must have gotten 22 from the father. That eliminates male number 1, but we already know that from the first location. Another X. Now, TH01. Mother has 4 and 9, and child has 4 and 6. Clearly 4 came from the mother, so 6 must have come from the father. Only male 2 and male number 3 have the 6 allele, so we can cross male number 4 off the list. He is not the father. 2 more X's. Finally, TPOX. The child appears to have 2 6 alleles. The mother has 1, but the father must have provided the other. Only male 1 and male 3 have a 6 allele. Put in the last X's. And we're left with male number 3. Is he the father? Well, we would want to check the other 9 locations. If he matched there, then we can be pretty certain that he is the father. To summarize, the sizes of fragments, RFLP or PCR, are affected by number of repeats and possibly mutation as well. VNTR is used to identify potential crime suspects and to determine paternity. PCR is crucial. It really speeds up the process to allow specific DNA regions to be examined. In the United States, CODIS locations are used to analyze cases and to collect data. DNA fingerprint databases can allow police to search for criminals who may match a particular allelic pattern. That's it for now.